Hello, good afternoon once again. I am Dr. Jose M. Oclarit, PhD in Applied Biochemistry and Molecular Biology at Hiroshima University in 1994. In 1990. 1990. I started 1990. I finished it in 1994. And in 1996, I was awarded as the most outstanding young scientist of the Philippines in the field of biochemistry by the National Academy of Science and Technology of the Department of Science and Technology in Manila. At present, I am a professor of medicine and biochemistry at the Matias H. Asnal Memorial College of Medicine, the first medical school outside Metro Manila, started in 1947. So right now, I'm going to post some statements regarding those who question whether or not the human cell has a reverse transcriptase. Indeed, there is a reverse transcriptase in a human cell. However, that reverse transcriptase is a special type of reverse transcriptase that is usually used to extend the telomere sequence. So that is the telomerase enzyme. I do not know, I'm not very sure also whether this can be used for the cell to revert the mRNA of the virus into a DNA. So if there is no evidence, I would like to stand corrected regarding that. However, there is indeed a reverse transcriptase in the human cell. Now, with regards to the infectivity of the virus, there are two general types of infectivity of the virus. One infective mechanism of the virus is what you call the lytic cycle. What is the lytic cycle? The lytic cycle is like this. The virus will first recognize the membrane of their living host. And once they recognize the membrane receptor of their living host, they will bind to it, like the spike protein, for example. The spike protein is used by the virus for it to be able to invade and infuse their genetic material to the host cell. So once they are inside the host cell, they will use their own machinery, or rather they will use their own genetic material to dictate the host to produce all the body parts of the virus. Like once they get inside, they will activate in the lytic cycle, they will activate their mRNA for the reverse transcription. They will produce an mRNA, or rather they will produce a protein reverse transcriptase in order to encode their RNA molecule into DNA and insert it into the DNA of the host. But that is not yet the lytic cycle. The lytic cycle is once the mRNA of the virus gets inside the host, they will now manufacture all the parts of the virus, and they will replicate everything, all the body parts, their capsid, their nucleoid protection, their protein, their genetic material, and they will assemble their body parts inside the host, and then they will multiply and multiply until the host cell is saturated and causes the lysis. That's why it's called a lytic cycle. And the lysis is now the time by which the virus can now infect other cells, or they will now, the lysis part there is the part by which the virus can now be distributed to other parts of the body, or other parts of or to other persons. You call it the shedding off. Actually, you call it the shedding off when the host cell is already lysed or lysed. That is what you call the lytic cycle of infectivity. The other type of infectivity is what you call the lysogenic cycle. The lysogenic cycle is the one that has the ability to revert their RNA material into DNA using their own enzyme that they have produced inside the host cell, reverse transcriptase, and then this reverse transcriptase will now be used to revert their RNA genetic material into DNA, and they insert it somewhere along the DNA of the host cell, and it will be there forever. That is why some of the oncogenic virus that, or rather some of the oncogenic genes that are found in human are actually introduced by this virus. You call it the oncogenic virus. That's why we do not know. We have already here the oncogenes that is introduced by the virus using the reverse transcriptase. That's why the virus is actually a very useful vehicle, because they can carry their genes, they can insert it inside the host. That is the ability of the virus. That's why it is being utilized and exploited by the molecular biologists to deliver the genetic material. That's why in the AstraZeneca vaccine, and as I was told, the AstraZeneca as well as the Johnson & Johnson, they are using what you call the DNA or adenovirus. And they are using the adenovirus of the chimpanzee because it does not replicate, but it can deliver and insert the genetic material to the host. That is what you call lysogeny. So there are two recap. So two types of infectivity: the lytic cycle that will enable the virus to lyse the host, and the lysogeny cycle, which is actually enable the virus to insert their own genetic material to the DNA of the host. That's why you become transgenic in nature. All of us are transgenic in a way because we are transgenes from our parents, but there are environmental materials like transposons, for example. Transposon is a jumping gene, and it can insert any genetic material anywhere, somewhere in our own DNA. And virus behaves like a transposon. So that is the real meaning of what we call lysogeny. So the mRNA vaccine and the DNA vaccine is dangerous because once is it there, it will be there forever, and it will reside on your own body through your cells forever. Thank you very much and good afternoon.